Whenever we pop a pill or get an injection, we usually just kick back and wait for our symptoms to clear up. But what's actually happening inside? At a microscopic level, that little medicinal molecule is embarking on this absolutely treacherous, highly complex obstacle course once it enters your body. Today, in this explainer, we are going to act as the ultimate tour guides and trace this incredible journey.
But before we follow our tiny hero, we need to map out the territory. We're operating in the wild realm of pharmacokinetics. If you break that word down, it literally translates to the quantitative study of drug movement. We're talking about how it moves in, through, and eventually right out of the body. Simply put, it's the science of what your body does to the drug.
To make sense of all this chaos, we're going to follow our drug step by step through 5 main checkpoints. 1. The drug's invisible journey. 2. Crossing the cellular border. 3. Absorption and bioavailability. 4. The distribution highway. And finally, 5. Navigating special body barriers. Let's get right into it.
Checkpoint 1. The drug's invisible journey.
Okay, so every single drug has to follow a distinct chain of events to actually be useful. It all starts with the drug in its dosage form, like a tablet. It gets released and dissolves into a solution in your system. From there, it's basically a 4-part dance. First, absorption into the blood. Second, distribution to the site of action or into storage tissues. Third, biotransformation, which is where the body tries to metabolize or break it down. And finally, excretion, flushing it out through urine, bile, or sweat. It's a continuous, dynamic flow from start to finish.
Moving right along to checkpoint 2, crossing the cellular border.
Now, to get literally anywhere on this map, our hero first has to get past the body's microscopic bouncers, biological membranes. There are 2 main ways our drug gets past the security. On one hand, we have passive diffusion. If a drug is lipid-soluble, it essentially acts like a ghost, passively slipping right through the cell's lipid bilayer from an area of high concentration to low concentration, no energy required at all. But on the other hand, we have active transport. Some drugs just can't diffuse. They essentially have to pay a VIP fee using the cell's ATP energy, binding to specific carrier proteins that actively pump them right across the border.
But wait, that brings up a really great question. You might be wondering, how do massive molecules, like huge proteins or complex hormones, manage to cross these rigid borders? I mean, they can't just passively diffuse, and they are way too big for standard protein carriers to pump them. So what happens?
Well, for the big stuff, the body uses something called vesicular transport. It's this fascinating 3-step process. First, the massive molecule binds to a surface protein. Next, the cell membrane literally folds inward, physically swallowing the molecule to form a tiny pocket, or a vesicle. Finally, that little vesicle travels all the way through the cell and is extruded or basically spit out on the other side. It's a microscopic cellular ferry system.
Which brings us to checkpoint 3. Absorption and bioavailability. Once we're past those initial cellular borders, we enter the actual process of getting the drug from wherever it was administered straight into the systemic bloodstream. But here's the kicker. A drug's absorption is absolutely not guaranteed. It relies entirely on a really specific set of physical and chemical factors. Take the area of the absorbing surface, for example. Your small intestine has a massive surface area because of these tiny finger-like structures called villi, making it a prime location for drug absorption compared to, say, your stomach. Blood flow or vascularity also plays a huge role. More blood flow means the drug gets swept away faster, keeping the concentration gradient steep and keeping that absorption moving quickly.
And understanding all of this is crucial for seeing why different drug formulations matter so much. If we look at a plasma concentration curve comparing 3 variations of the exact same amount of a drug, the differences are wild. Formulation A spikes super quickly, shooting well past the therapeutic concentration line, meaning it gets absorbed incredibly fast and works fast. Formulation B, though, it's absorbed much slower. It has a flatter curve, but it actually stays in the therapeutic zone for a lot longer. And formulation C barely reaches the line at all. So how a drug is literally manufactured completely alters its absorption profile.
This perfectly introduces the concept of bioavailability. Think of bioavailability as the actual fraction of the drug that makes it into your systemic circulation completely unchanged. This is exactly why some medicines just have to be injected directly into a vein. An IV injection gives you 100% bioavailability. Boom, it's all in the blood. But if you swallow a pill, it might not be completely absorbed, or it might get destroyed by your gut wall or your liver before it ever has a chance to reach your wider bloodstream.
All right, on to checkpoint 4, the distribution highway.
So, assuming our drug has successfully made it into the blood and has officially merged onto a massive distribution highway that connects to the rest of the body, there's this really clever pharmacokinetic concept called the apparent volume of distribution. Basically, scientists pretend the whole body behaves as a single uniform compartment. Say you inject 1,000 milligrams of a drug. Later, you test a sample of blood plasma and find 50 milligrams per liter. You do the math and deduce the drug is distributed across an apparent volume of 20 liters. It's a neat trick that helps us understand if a drug stays mostly in the blood or if it spreads deep into the surrounding tissues.
But here's the thing. The drug doesn't always ride alone on this highway. Many drugs actually immediately bind to specific plasma proteins. Think of a protein like albumin as a biological taxicab. Acidic drugs like penicillin or ibuprofen bind heavily to albumin. And once they're buckled into these large proteins, they are essentially trapped inside the vascular compartment. They literally can't cross capillary membranes to do their job until they unbind. It creates a sort of temporary storage system circulating right inside your blood.
Even wilder, drugs can leave the blood and get sequestered or permanently stored in specific tissues. Heart medication like digoxin actively concentrates in skeletal and heart muscle. Antibiotics like tetracyclines can actually lock right into the calcium in your bones and teeth. And highly lipid-soluble drugs like some anesthetics can literally dissolve and hide out in your body's fat stores, meaning they can hang around in your system for a very, very long time.
And finally, checkpoint 5, navigating special body barriers.
For our final chapter, we have to look at where the drug can't easily go. The absolute most heavily guarded fortresses in the human body. Treating neurological conditions like Parkinson's or meningitis is incredibly difficult. Why? Because the brain is a true VIP organ, and it all comes down to the microscopic architecture of the blood vessels up there.
Let's dive into this stark contrast. Normal capillaries throughout your body are, well, kind of leaky. They have these large spaces between the cells that let almost anything filter through. Brain capillaries, however, they are locked down tight. There are literally no spaces.
The legendary blood-brain barrier operates on a strict lockdown. A usual capillary is porous, sure, but a brain capillary has tight junctions sewing the endothelial cells completely together, plus a whole extra layer of glial cell processes wrapping around the outside. It forms this impenetrable wall against potentially harmful, non-lipid-soluble substances. Only highly lipid-soluble drugs, or those with very special transporters, can actually sneak into your brain.
Conversely, and this is absolutely vital to remember, the placental barrier during pregnancy is very different. It is highly lipodal, meaning many lipid-soluble drugs taken by a mother can very easily gain access to the fetus. It is an incomplete barrier. This single pharmacokinetic fact is exactly why doctors have to be so incredibly careful about which medications are prescribed during pregnancy.
As we wrap up our hero's journey today, this quote from the source text really hits the nail on the head. It perfectly summarizes the sheer chaos of pharmacokinetics. Distribution isn't just about dropping dye into water and watching it passively spread. Human biology actively wrestles with the medicines we swallow, binding them, moving them, trapping them, and filtering them every step of the way.
Now that you've seen the incredible, treacherous obstacle course playing out inside your own cells, it kind of changes how you look at a simple pill, doesn't it? Consider how this knowledge of membranes, bioavailability, and cellular barriers will shape the next generation of targeted medical breakthroughs. How do we design a drug that actually tricks the blood-brain barrier? How do we stop drugs from hiding in our fat tissues? That is the absolutely fascinating frontier of pharmacokinetics. Thanks so much for joining me on this explainer, and keep exploring.